Dr. Khetarpal, shall we begin?
Yeah,
Yes,
we have
if I
more
suggest,
than 300. I think we
if I
should.
suggest something before we start, the organizer can actually mute all the participants. And
Okay.
We are doing that, sir. We are about to do that. And
Okay.
one more request, Dr. Jimmy, if you can, since I sent you my link to everyone, if you can change your profile name, it's showing my name. So just right click and change it to your name. Okay, I think we will start. A very good morning to all, and good afternoon and good evening wherever you are joining for this webinar. And welcome to this first webinar on genome editing tools and its applications for targeted plant breeding, which is a part of the webinar series that APARI, KBCH and BCIL are jointly organizing, and with the topic applications of genome editing for sustainable agriculture and food security in Asia Pacific region. As I mentioned, this is being organized jointly by Asia Pacific, by APARI, which is the lead organization, jointly with Korea Biosafety Clearinghouse and the Biotech Consortium India Limited from India. So this is actually, as I had indicated in my invitation letter to all the participants in our webinar announcements, that this proceedings of this webinar are going to feed into a resource document which is being prepared under the guidance of a very senior level expert committee, our steering committee, and I would like to place on record our thanks and the guidance which they have provided in planning and undertaking this webinar series. I would quickly give the names of our steering committee members, who are the senior academic experts as well as the members of the regulatory committees in countries in the Asia Pacific region. So these are Dr. Norvati Adnan from Malaysia, Dr. Flarida Carino from Philippines, Dr. Homin Jain from Biosafety Clearinghouse, Dr. Ryo Osawa and Dr. Manaba Takahara from the University of Tsukuba, Japan, Dr. Maureen McLean from the United States, Dr. Heidi Michel from Australia, Dr. Roland from Taiwan, and Dr. Kiran Sharma from Equisat India, and Professor Yang. And I also welcome my colleague, Dr. Rishi Tyagi from APARI, who is also the co-convener for this whole events. So with this brief background, I would like to share with you also one more very good response that we got for this webinar. There has been a lot of interest in the topic, especially from the Asia Pacific region, and I'm pleased to share with you all that we got almost 2,000 registrations, and very pleasantly I'm sharing that from almost 60 countries, not only from the Asia Pacific region, but around the globe. Just brief information about the webinar. I would just like to also request everyone that please keep yourself mute. Of course, the organizers are keeping the mute, keep everyone mute, and post your comments, remarks, questions, everything in the chat box. We are, depending on the availability of the time, we are going to try to answer the important questions. However, if we are not able to give any response, we are going to talk to the speakers, the relevant speakers, and individually get back to you on all your queries, comments, etc. With this, I will close, and I would like to invite Dr. Ravi Khetarpal, who is the Executive Secretary of the Asia Pacific Association of Agriculture Research Institutions, that is APARI, the lead organization which is leading this effort, to kindly give the welcome remarks and the introduction. And I would also like to thank Dr. Khetarpal for his very active involvement and the support in taking forward this initiative. Dr. Khetarpal, sir. Sir, you have to unmute yourself. Sir, you are on mute.
I'm sorry for that. Okay. Thank you, Dr. Vibha. And welcome to all the participants once again. I have a pleasure and privilege to give a welcome speech on this very important webinar series, which APARI is co-hosting along with Biosafety Clearinghouse of Korea and with Biotechnology Consortium of India Limited, as Dr. Vibha just mentioned. Well, I'll be very brief and I'll just give you a bit of a background to set the stage. As you all know, Asia Pacific region is struggling hard to achieve sustainable development goals of United Nations. And we are lacking far behind as per recent report from United Nations Economic Social Commission for Asia and Pacific. And we are not even halfway through of achieving our target as of now. COVID-19 has compounded the situation further. Also, Asia Pacific is having the largest chunk of demographic or the population. More than 60% of the world population lives here. Hence, the challenges are in abundance. Now, in that context, I will tell you in brief about APARI and who we are and what we are doing. We are a membership-based organization established way back in 1990 under the aegis of FAO of United Nations and has a mission, a clear mission of strengthening agri-food research and innovation in the region for sustainable development. We have more than 80 members, which include country members, international research organizations, education institutions, some private sector organizations, and also sub-regional fora like SAARC in South Asia or SPC in the Pacific. We work on bioresources and biotechnology through our program on Asia Pacific Consortium on Agriculture, Biotechnology, and Bioresources, which is led by our coordinator, Dr. Rishi Tyagi. We also work on risk mitigation, where we work on the WTO projects for compliance to SPS in Southeast Asia and in South Asia. We work on inclusive development for pro-poor value chain, and we definitely work on policy and advocacy. Through our programs, which are on knowledge management, on capacity building, foresight and visioning, including women and gender issues. With that background of Asia and of APARI, of Asia Pacific and APARI, I would like to say the challenges in agriculture, research, development, and innovation before all of us are many. And some of those challenges, as you all may know, are surmountable where the technological innovations are concerned. But there are natural calamities and disasters which are unforeseen and other areas where we all have to live with the mitigation strategies as much as we can. In the field of biotechnology, there have been challenges all through, from the birth of biotechnology, from the birth of genetic engineering from 1990s. There are challenges right from investment up to the application and some complications of the technology. Hostile public backlash, especially in European Union and other parts of the world on GM crops, all due to ethical considerations from consumers' angle. In the context of this background, I would say gene editing has come up as a big promise, as an important development that holds promise to advance our agricultural sustainability and nutrition security, where extrinsic DNA sequences are not involved, as in developing GM crops. We have a rich pipeline of gene-edited plants which are being developed both by public and private sector organizations across the Asia Pacific. Application of gene editing is receiving a lot of interest in the last few years. And with the large-scale commercialization which has to be done on gene-edited plants, important events have been organized and address the policy and regulatory issues. The one which was organized recently, 2.5 years back, was Second Asia Forum on Genome Editing held in Gangwon in Korea. And as a follow-up of that, later we, APARI, through our program of EPCORP, along with ICRISAT, convened the regional expert consultation on gene editing and its regulation in about 1.5 years back in October 2019. The expert consultation resulted in series of recommendations relevant to any potential regulation of gene-edited plants. In fact, what APARI is doing is not only organizing events and workshops and expert consultation. We really mean the recommendations of the workshop, and wherever we can move as much as we can from APARI's secretariat side, we try to carry it forward, though there are many things which the National Agriculture Research System has to take up. Now, APARI, in the light of recommendations of APARI ICRISAT expert consultation in October 2019, we initiated the exercise of the development of a comprehensive resource document on application of gene editing in sustainable agriculture and food security in Asia Pacific region. This, we hope, will provide government and other stakeholders with information needed to take a policy standing or a policy positioning as regards to issues related to any potential regulation of gene-edited plants. In this endeavor, I am happy to say we have developed, along with the steering committee whom Dr. Vibha warmly acknowledged, the outlines of a document that will cover key issues relevant to research, development, and deployment of gene editing in plants in agriculture. We have identified the lead authors, and we are hosting a series of webinars to solicit inputs from all of you, the participants who are the key stakeholder group, on the topics which are listed in our resource document to be published. We will see how best we can diffuse the document once it has come up in the coming few months through a very effective communication plan. And then we intend to organize a high-level policy dialogue, a high-level meeting to ensure its adoption at different levels in different countries of Asia Pacific region. I hope you will participate actively in the webinar being moderated by Dr. Ryo Osawa, a well-known biotechnologist from Japan, and enjoy deliberations by excellent speakers and panelists from Japan, Australia, Bangladesh, Korea, Malaysia, and Taiwan. Before I conclude, I will profusely acknowledge the support of KBCH, both technical and financial, and of BCIL for collaborating actively with APARI, and to all the participants involved in the webinar. And my special thanks again to all the esteemed members of our International Steering Committee who carved the roadmap of webinar series, for initiating the exercise of reviewing the genome editing tools, its application from lab to line, and enabling policy positioning. Thank you, one and all. Over to you, Dr. Vibha.
Thank you so much, sir. Thank you for the elaborate introduction to the whole concept and why we are all here to listen to our esteemed experts. Without wasting time, I would like to now introduce Professor Ryo Osawa, the chair for this webinar Professor Ryo Osawa is the Dean of the Faculty of Life and Environmental Sciences at the University of Tsukuba. And he has both the research and the regulatory experience being associated with the regulatory system in Japan. And we've already circulated the profiles of all our speakers to the audience, and that's why I won't go too much into detail and would request Dr. Osawa to kindly give his remarks and carry on the proceedings of the webinar. Thank you very much, sir. And we also wish to, I just also want to say that Dr. Osawa is very busy, and sir, we are really, really thankful to you for kindly agreeing to be with us and, you know, share, be part of our endeavor.
Okay. Can you hear me? So thank you very much for my introduction. So Dr. Bibha. So thank you all for joining us today. So looking at the poster of webinar first, it looks as if I'm holding down the presenters. So it's my picture is top. So I feel that my face is a little bit larger than the others. I'm afraid, but I'm not only just moderator for today, okay? Not regulator. In Japan, I'm in charge of regulating the products of new gene technology. So that's sometimes the enemy of the Professor Ezra. So, but I am not here today to regulate the speech of the presenter, of course, yeah? So I moderate the speech. It all starts with scientific understanding is very, very important, I think. I'm looking forward to working with the participants to gain a deeper understanding of new gene technologies and in each situation in each country from the panelists. So Dr. Bibha said to me, so before this meeting, that how many people joined this meeting? So more than 1,700, near the 2,000. So this news is completely, I'm freaked out, sorry. So that's a rapid, I think so, okay, it's a joke. So I think the rapid pace of the developments, very, very rapid pace of the developments are taking place in application of gene editing in agriculture for research and commercialization at global level, as you know. So, but Asian countries also will naturally have to continue our sustainable agricultural production in this web, within this web, okay? So today's these webinars, so Dr. Rabi already introduced. So we prepared. Object is to discuss how gene editing can be effectively used for sustainable agriculture and food security in Asia Pacific region. Okay, so it's my job to give the presenters more and more time. Sorry. So today we have two distinguished scientists giving presentations. First, Professor Ezra from Japan. Here's my colleague. So we'll introduce a case study on how germinating breeding countries to human health contributes to human health. And next speaker is Professor Botella, will introduce a significant contribution of germinating to crop development. After that, we will ask the participants a few simple questions by using Zoom poll system. So please answer them. This is read by the Dr. Bibha. So this will be followed by panel discussion, okay? We have invited scientists from 5 countries. 5 countries, right? Okay, maybe, yeah. So to participate in the panel discussion, I would like to introduce them again at the time. So I would like to introduce the first speaker, Professor Ezra. So the Dr. Ezra from University of Tsukuba the scientists with interest in molecular breeding of Solanaceae and Cucurbitaceae. Solanaceae, mainly tomato. Maybe you can, everybody can see his back. Maybe the tomato, yeah. So, and Cucurbitaceae crops. His major interests are research and development of biotechnology, including the cell tissue culture, GM crops, and gene-edited crops. He led a Japanese national project on application of gene editing technology to agricultural crops. So he has successfully developed the GABA-rich tomatoes using the CRISPR system. In 2018, he founded university venture company, named Santec Seeds, where he currently is maybe the chief technology officer. Yeah. So in Japan, the first product to complete the voluntary notification process of the Ministry of Health, Labor and Welfare, or the Ministry of Agriculture, Forestry and Fisheries, to verify whether germ-edited products should be regulated as gene editing products is a GABA-rich tomato. Yeah, sorry, gene engineering products, sorry, GMO. So last year, the both ministries announced their decision that germ-edited tomatoes will not be regulated as genetically modified products. I think so. This is a landmark event for Asian countries. For more details, maybe let's hear from the professor as well. So this is, I think, this is good news for not only Asian people, not for also for the world, I think. So let's hear from Professor Ezra. Okay, Professor Ezra, please.
Thank you very much for the introduction, and I would like to appreciate the organizer to invite me to give a chance to introduce our recent achievement in the application of gene editing technologies in the crop improvement. Can I share my slide? Okay.
Yes, sir. Please.
Okay. Can you see it?
Yeah. Can you make it in the presentation mode, sir? Yes.
I changed it. Now it's okay. You can see it.
Okay, no problem.
Okay, so thank you very much for the introduction and invitation to this important webinar series by APARI. And today I'm going. Okay, so today I'm going to introduce the application of gene editing technology for the application of tomato and nutritional quality, which is because today the topic is application of gene editing in plants for agriculture, sustainability, and nutritional security. So my topic is a case study of a nutritional security establishment. Okay, so in this first slide, I would like to a little bit introduce the how we developed this new gene-edited crop. And in this slide, I would like to emphasize that the government support is very, very important to introduce new techniques in the society. So in Japan, we learned the national project, which is called the Cross-Ministerial Strategic Innovation Program, Promotion Program, just SIP project, which is supported by Cabinet Office, Japanese government. And in this project, we try to demonstrate or apply gene editing technology for the improvement of agricultural products, including cereals and fruits and some potato and some other fishery products. So in this project supported for 5 years from 2014 to 2018, and in this project, I established 4 groups, which one group 1 and group 2 and 3 and 4. And the group 1 is to establish a new or improve the gene editing technology because of that 2014 the CRISPR-Cas9 technique has just released after 2 years. So that's why we try to improve the techniques more friendly to the application of gene editing application to agriculture products. And also we also try to find the target gene of gene editing. And also using the technology and the information, we try to demonstrate actual products by using gene editing technologies, and which include tomato, rice, potato, and tuna fish, and others. And also the unique point of this project was we also simultaneously started the public communication with the consumer or different stakeholders to introduce or get increased understanding of this new technology, which is led by the Ryo Osawa, today's moderator. And so I spent 5 years. And finally, eventually we developed a potential crops improved by gene editing crop and gene editing technologies. In this slide, you can see some example, not all, but some example. Now we try to improve the rice yield. Then we could increase the 20% of yield by gene editing by knocking out the 1 genes. And also we try to reduce the natural toxin in potato. Then solanine and chaconine is a famous natural toxin included in the potato. By knocking out the biosynthetic gene related to these toxins, that we could reduce significantly reduce the amount of natural toxin in potato. And also then we try to improve the tuna fish, which is easy aquaculture for easy aquaculture. And my group is focused on the improvement of vegetable crop, mainly tomato. So in this project, I think that the motivation is very, very important to introduce this kind of new technology to our society. So in this here, I'd like to show the situation in the population changes in Japan. And every people say, and the scientists say that the world population is going to increase up to 2050 to almost 10 billion. But in Japan, we already, the population peak was 2010, and that's after we are losing a population gradual, step by step, as you can see here. So, and then we are facing the national issues based on this population decrease. Okay, and here you can see the changes of population structure in Japan from 1975 to 2050. Now we are around 2025, and 50 years ago we have a lot of young generation. So then Japan is very, very active. And then in 2000, we had a lot of middle ages population, and now we have a lot of aged population. During the and we are expecting the more than 40% of Japanese population will be more than 65 in the 2050. And these population changes bring us the changes, the increase of lifestyle-related diseases. And this one is also by the change of a food habit. And so then, but anyway, so the health promotion by daily food is a very important issue in our countries. And I think that these changes will be same in other Asian countries where the population is growing and also start reducing. So maybe we are facing same problem for the health. And so that one of the most comprehensive method is to use a healthy food through daily consumption. So that's why we decided to improve the nutritional quality of the crops. And in this study today, I would like to introduce the nutritional alteration of this by gene editing techniques. at this moment, my group is trying to understand the important breeding trait, the molecular mechanism of the important breeding trait, like the shelf life and fruit setting, and the sugar accumulation and the nutrition. But today I have 20 minutes, so that I just focus on the improvement of nutritional quality in tomato by gene editing technologies. What is a GABA? GABA is a very famous in Japan now, which is a functional nutrient which can promote the human health. And this GABA is a gamma aminobutyric acid and abbreviated GABA. And it is involved in the biotic stress responses in different organisms. But for human, it has a function as a neurotransmitter that's resulting in the reduction of blood pressure and also induction of relaxation. Because of these functions for human, and many Japanese food companies is using this GABA to their food products. And recently, more than almost 500 GABA-added food has been commercialized in Japan. That is very famous. And that means that this GABA is a very famous functional ingredient for Japanese consumers. So tomato is one of the vegetable which highly accumulate GABA. And then initially we spent a lot of time, almost 10 years, how to understand how the tomato accumulate the GABA in the fruits. In the basic research showed us that the GAD3, which is genes responsible for the GABA biosynthesis. Tomato has 5 GAD genes in the tomato genome, and I look at the tomato genome, but the GAD3 is predominantly expressed in the fruits, and which is regulating GABA accumulation in the tomato fruits. So that's why, and then we got such information, and then further we did some biochemical analysis. When we see that this enzyme, we could find small tails at the C-terminal end of this protein, which is a OD inhibitory domain, AID domain, and this short tail replace the enzyme activity of this enzyme. So then we deleted this tail, and then I expressed it in a tomato, and then so that's a enzyme produced from this modified gene. They produce a show a high GABA activity and resulting in the GABA accumulation. Based on basic research, we try to create the same mutation by gene editing techniques. And in this study, we use a CRISPR-Cas9 system, which is a very famous genome editing. So far, there is a many kind of gene editing technology, like first generation, second generation, and third generation, and even more fourth generation. Our this CRISPR-Cas9 system is a third generation gene editing technology, which is most widely used in different organisms, not only plants, to animals and some other organisms. So we decided to use this CRISPR-Cas9 system to make a mutation. So we introduced a mutation, just in stop codon mutation, created a stop codon mutation just in front of this AID domain, and then we cut off the tail from this enzyme. Then, as expected, we could introduce a stop codon mutation in front of AID domain, and this is a different line we created. And then when we measure the GABA accumulation in the in those mutants fruits, and here is the GABA content of wild type, but the mutant shows higher GABA accumulation, as you can see here, and so that's why this is as expected. So then after getting this result, we used because the tomato has been used as a F1 hybrid plant. So then we created the high GABA tomato line created by gene editing, and other parental line make a F1 hybrid. And we expected that this high GABA trait must be a dominant trait. And then we checked the GABA accumulation in the F1 hybrid line. And this slide shows the GABA accumulation. the white bar shows the control line. These other two lines show the F1 hybrid line produced by crossing the high GABA tomato line created by gene editing and other pure line. And then we measure the GABA accumulation at the different stage of fruit development, mature green stages, breaker stages, and late life stages where we can harvest the fruits. And then in these late life stages, the fruit, F1 hybrid line created by high GABA line show a higher GABA accumulation. It's a 4 to 5 times higher GABA accumulation than the control line. So, from this is these are proof of concept POC experiment, and then we know that the by engineering GAG3 genes, then we could increase the GABA content. So, and after that, we try to develop an actual commercial line using these techniques. But for eventually we wanted to commercialize our products. So that's why to get the CRISPR license, we established a venture company. The name is Sanatex Seed, which is a spin-out venture companies from our university. And then I established the companies in 2018 to demonstrate the use of to demonstrate and create the high GABA tomato line for commercialization. And then here this slide shows that some patent related, and the CRISPR-Cas9 patent is basic patent is holded by 3 organizations. Here you can, as you can see here, but the patent in the CRISPR-Cas9 in plants is not so complicated like in animal. Animal is very, very complicated. But the plant is very simple. And companies at the time the name was DuPont, but now it's changed to Corteva. This Corteva accumulated these 3 basic patents. So then just we need to discuss with Corteva how to use. Then this Sanatex Seed company discussed with Corteva and finally obtained a license for commercialization of this product technology. Okay, and then so using the technologies and the patent, we created a new tomato cultivar based on the leading tomato cultivar sold in Japanese market. And the name is the original cultivar is the name is a Sicilian Rouge. This is the original cultivar. Then we engineer the 1 parental line for increase the GABA accumulation. So then finally we established a Sicilian Rouge high GABA. This is a commercial name. And then we started the commercialization in May 12 this year, just 2 months ago in Japan. And here's in this slide, as Ryo explained, in Japan, to deregulate Japan, we need to notify the products to 2 ministries. Ministry of Agriculture and Forestry and Fishery and the Ministry of Health and Welfare. So we did, according to the Japanese system, before notification, we need to pre-consultation with 2 ministries. And we did that pre-consultation with some data. And in Japanese system, we have to provide some data related to our products. Then we submitted our data. And then we get some pre-consultation with the 2 ministries, and it takes almost 1 year. And then last December, we could complete the pre-consultation, and then we could submit the notification, and then they accept our products. So then we submitted for our cultivar for cultivation and food and feed. So now we can, our Sicilian Rouge high GABA tomato, we can use it as a normal tomato, that we can grow it anywhere in Japan. And then according to the development, during the development, we made a discussion with different stakeholders, including consumers and the producers. And because this is the world first CRISPR crop commercialized in the world. So that's why the consumer have some concern, and also some stakeholder. So that's why they need the information. So as a initial products of this technology, we decided to, the company decided to provide the information, and that's means going to labeling. And then we are going to provide the information to the consumers using this labeling, this label, and which contains the information of this tomato has been improved by gene editing technology, and a notification to MAF and MHLW has been completed. So, and then we finished the notification last December. So then that after we wanted to show our or feel our products by the consumers or users. So that's why we decided to freely provide our the gene edited tomato to the home gardeners, because the home gardener can they can grow and they can eat, and they can harvest and they can eat, cook and they can eat by themselves. So then we announced that we are going to provide a seedling, free seedling to the monitor. Then surprisingly, we had more than 5,000 applications. Last from last December to this February, just 3 months, we got more than 5,000 applications. And then we provided the seedlings, as you can see here, as you can see in this slide. And we started delivering this seedling this May, and we provided this seedling to all over Japan. And then the home gardener started growing, and we had a lot of information that they started harvesting and enjoying this high GABA tomato. So here I'm also growing my tomato in my garden, and I start the growing in the June 2nd, and then June 16th we got start setting, and then just yesterday I harvested the fruits, as you can see here. So looks so Japanese gardeners enjoy the growing and harvest, and also tasting these products. Okay, so I think this is a final slide, and this is my message. The new era of the rapid and precise breeding via gene editing technology is coming. So that's GE allow us an order-made breeding for a sustainable and a nutritional food production, which as introduced in my presentation. Okay, thank you very much for your attention. I think that's all.
Okay.
Okay.
Thank you very much, Hiroshi. So it's thank you very much for this very useful talk to the participants. And I would like to take a few questions. So please use the raise hand function in Zoom, or I always send through the chat if you have any question in the chat box. Okay. So sorry, I cannot follow the hands. Sorry, with many people. Okay. Any questions? So okay, from the chat box. So okay, so the very simple question I have. So for example, so that this Sicilian Luz high GABA tomato is regulated in Japan, and how long it takes time for approval process. And I maybe confused. There's no regret in Japan.
Okay,
right?
so and I started Japanese regulation rule was established in 2019 October. So then we started the pre-consultation with 2 ministries, as I explained, and I spent almost 1 year to complete the notification. So that in this case, the answer is it takes 1 year to complete the notification.
Okay, I know well. So that is process. So it's very shortened. So I think so. But every consumer party says it's a little bit too short.
Yeah.
Yeah, so that is a very, very good consulting. So have done, I think so. Okay, so any other questions? So from the Clement from Malaysia. So may I know whether the post-harvest sensory quality of tomato fruits are affected using this technology? So maybe the latest GABA tomato is not related, I think. So related?
Yeah, okay. So the accumulation of GABA is not so effect on the post-harvest performance. And because we increased just 4 to 5 times, that is enough for getting the expected functions of this amino acid. So, and when we compare the original cultivar and the engineered line for the shelf life, and there we could not change, or maturation, we could not change, detect any differences for the maturation and also shelf life between the original line and this engineered line, even if we increase GABA accumulation in the fruits.
Okay, so another question is related to your question answered. Do you have any plans to commercialize and sell outside of Japan?
Okay,
It's maybe the secret of your company, yeah?
No, not that a secret, because that I think that like high blood pressure is a kind of a national project in the national program in many countries. So if the regulation is become established in other countries, and also the maybe harmonization, similar regulation rule established in the some other countries. So then we would like to share our achievement with those countries.
Okay, thank you. So this question is very important. So, and the answer is also the important. So harmonization is very, very important to export and also the import. Maybe this problem will be discussed the third webinars in this series. Maybe.
Yeah.
Thank you. So another question. So it's a very difficult question. So can you describe the public acceptance of gene-edited plants, fruits, vegetables among the Japanese consumers? So how to do that so that get the acceptance of gene-edited plants in Japan?
Okay, so that's a very good question and a very difficult question to answer. And as you know that there is many people they have a different opinions, and so I think that it is very different.
Gulot ako yung RK ko. Tapos ako pa naririnig.
Please, somebody, please mute.
Okay, okay, can I continue?
Okay.
Okay, that's a very important issue too. But as I introduced, we discussed how we can provide these products to the consumers. So, and if we use normal pathway to the consumer, like farmers produce fruits and vendors can collect the food, and those route is quite difficult to get initial products. So then we decided to developer to consumers route. That means that using a SN network web, then we decided to provide our products to the consumer who really wanted to this one. So this is, and then actually I introduced, we have more than 5,000 gardener consumers that are interested in these products. And then maybe in the next year, they will release the information to their friends, so that this 5,000 increased to maybe 50,000. And then the next year, maybe 5 million people interested in this.
Yeah.
So that's why this is our strategy to get consumers understanding.
Okay, thank you. So it's not so time now. So, but the final question. So this is a technical question. How can we identify gene-edited crops do you any specific markers? That means so that this one asks you how identify gene edited crops, or how can identify to do so gene edited crops.
Yes, thank you very much.
Yeah.
In Japan, the mutants created by this technology considered to be same mutants with the conventional breeding techniques. So that's why in this the products we produce, we know the mutation point in the target genes. So then, if we have such information, we can detect this mutation, these cultivars.
Yeah.
So we are providing those information to the government already. So the government has the information. So then, if the developer notify correctly, so then the products can be potentially detected.
Okay, thank you. So the more than 30 questions with answer, yeah, from audience. So I don't have much time left. So let me introduce the next speaker, the Dr. Bibha. Okay, so I don't have much time left. I'm sorry. Let me introduce the next speaker. So Dr. Jimmy Butler is a professor of plant biotechnology at the University of Queensland, Australia. He is the head of the plant genetic engineering laboratories, so specializing in the fields of tropical and subtropical agricultural biotechnology. Yeah, so he has 11 international patents in the field of plant biotechnology and has founded 2 biotechnology companies. I'm very sorry, I cannot introduce the name of these companies. So I cannot only the scientific seeds. I'm sorry. So please introduce yourself, Jimmy. He is the member of the expert scientific panel for the Agriculture Biotechnology Council of Australia. So his research interests include biotechnological approaches for crop improvement, germ-edited plant pathologies. So as you know well, every participant know well, that traditional breeding for crop improvement is a very time-consuming work. The laborious and costly process is needed. Every plant breeding scientist said that CRISPR has changed everything, and also said the procedure is extremely easy to use. I can agree that. So very accurate and has already been used in a large number of species. And Jimmy will discuss the technical nature of CRISPR and describe a number of examples in which CRISPR has been used for ultra-fast breeding. So the ultra-fast breeding, so said by himself. Okay, so please, Professor Jimmy Butler, please.
Okay, thank you very much. Thank you very much for the nice introduction. I will share my screen now. Okay, I think that, can everybody see my screen now?
Yes, okay, no problem.
I take it that you can and that you can hear me. First of all, before I start, I would like to thank the organizers for this fantastic opportunity to participate in this forum. We do have been working with CRISPR for quite a long time from the very beginning. And today I'm going to give you a bit of a perspective of CRISPR, what is coming, and also a nice example that you can see from our own research that, you know, what CRISPR can do for you. To start with, let me start with mutation breeding. Yes, before me, you know, Professor Ezura, I was talking about, you know, the mutations that is, you know, no more than that, just mutation breeding. And that's what CRISPR is. Mutation breeding has been done for a long time in agriculture. And the funny thing is, almost 2,500 years ago, we have the first example of a mutant selection by humans. And it's in a Chinese book called Lulang. It was written in the 300 before Christ. And that mutation happens because of the sun. The ultraviolet, radiation from the sun produces mutations. Some chemicals produce mutations. Humans started actually inducing mutagenesis very early. In the 1930s, it all started. It didn't last for 1 or 2 years. It lasted till today. And today, we have more than 3,000 released commercial plant varieties, 3,000 around the world. And if you look at this graph over here, you will see that 2,000 of them, more than 2,000, are in Asia. So Asia is a major adopter of mutated plants, crops that have been obtained by mutagenesis. And why am I talking about mutation breeding? I'm talking about mutation breeding because genome editing, in a nutshell, is pretty much mutation breeding, but it has some small differences, or not so small differences, okay? Mutation breeding will create anything between 10,000 and 100,000 mutations. With CRISPR, you induce one single mutation. Mutation breeding is completely random, while CRISPR is extremely precise. Mutation breeding produce mutations anywhere in the genome, in any chromosome, while in CRISPR, you mutate one single location. And with mutation breeding, you need to produce hundreds and hundreds of lines to find one that you really like or that is useful, while for CRISPR, 4 to 6 lines is more than enough to find what you want. And finally, mutation breeding needs a number of generations because you need to backcross to the parentals a number of times till you weed out most of the mutations that you don't want. While in CRISPR, you can do it in 2 generations, and you will see about it in a minute. And again, let me explain that more than 2,000 mutated varieties are commercialized in Asia. So because I know that we have a mixed audience, I have prepared a very easy explanation what the requirements, what the CRISPR and genome editing is, concentrating on CRISPR. Genome editing is about producing a mutation in a very specific part of a chromosome. And we use a system that contains 2 elements, the molecular scissors and a homing device. Now, the molecular scissors is most of the time what we call the Cas9 enzymes. Well, not, there are other Cases, but most of the time the more popular one is Cas9, it's a protein. And the homing device is a small molecule of RNA. That molecule of RNA will guide these molecular scissors to the proper site, and that's what is called guide RNA. So let's say that you want to introduce a mutation in this particular chromosome, in this specific place in the chromosome. So all you need to do is design your homing device, and that homing device will bring the Cas9 into position, and the Cas9 then will actually cut the chromosome, produce what we call a double strand break. So what you have now is you have a break in your chromosome. And that is pretty common. That happens more times than you think. So cells actually have a repair mechanism that will come into position and will fix that break. Not a problem. 99% of the time, they come and they fix the break, and there is nothing strange in there. But sometimes that repair mechanism will come and will try to repair it, but will make a mistake. When that happens, a mutation is introduced. And that is how we create a mutation. But let me explain one thing. CRISPR is about all the way till here. It's about being able to cut DNA. The rest is completely natural. You just use the repair mechanism that exists in every cell, in humans, in plants, you name it. So one very, very quick example of how can you use CRISPR. This is a project that I had with while I was doing a sabbatical in Professor Yang Kansu's lab from the Chinese Academy of Science. And we asked ourselves, how can we use CRISPR to improve rice quality without affecting anything else? Just change the rice quality. But then when we started working, we thought that's a problem. because today there are more than 40,000 rice varieties that are grown all over the world. So you cannot really say, look, I'm going to change, rice quality, because there's no such a thing as one single rice variety. There are too many of them. So we change. Our question is, can we use CRISPR to improve the quality in local varieties, elite local varieties, without affecting anything else? And we chose two varieties that are pretty popular in the region where I was working, XS 134 and 9522. One of them is grown in the Jiangsu and Zhejiang provinces, as well as Shanghai. And the second one is grown in the south of the Jiangsu province. That gives you an example. Look, two varieties, and one of them is not even all over the province, it's only in the south of it. So it's important to be able to actually design and breed varieties for local farmers. So what did we do? We started working with it. We designed our target. This is the gene. And what we wanted to do is convert normal rice into sticky rice. Two different kind of rice for different kind of foods. But we wanted to see, can we just change the normal rice into sticky rice? There is one gene that controls that. And here it is the gene. This is the target that we have. And this is exactly, if you're looking here, this is the target that we have for mutations. And if you look at several of our lines in one variety and the second variety here, you can see that the mutations are extremely precise. They come straight right in the place that you want to do it, to the base, exactly in the place that you want to have it. So we produce a number of lines, different kind of mutations. Some of them had introduced one base, some of them had deleted one base, some of them have replaced one base, etc., etc. And then we started looking at the plants. Now, once we started looking at the plants, we grew them side by side. This is what's supposed to be the Woxie, means the sticky rice variety, and this is the original one. And if you look at them, they look pretty much the same. Then we look at the seeds. When we look at the seeds were, they have the same width, they have the same length, they were completely identical. And then we put it on the field. Actually, we put two generations on the field. When you put it on the field, this is the original, the parental variety. This is the sticky rice or the Woxie variety of rice. Absolutely the same yield for one variety and for the second variety. Nothing different. Now, you look at the total starch content of the grain, of the rice grain, you see the parental and the Woxie, the sticky rice varieties, the same amount of starch. So what did it change? It changed the composition of the starch. Woxie varieties or sticky rice varieties are high in amylopectin and low in amylose. This is the normal varieties, up to 20% of amylose. These are the CRISPR varieties, down to around 2.5 to 3%. And you can see it because these are the grain of rice. The Woxie varieties are a lot whiter in color. And if you stain for amylose, you see the normal parental, it stains pretty black because it has a lot of amylose. The CRISPR varieties are a lot lighter because they don't have that much amylose. Then we actually excise the gene by self-pollinating the heterozygous plants or back-crossing it with the original ones. For both varieties, we found plants that did not contain any of the transgene. Now, all that was done in the T2 generation. So yes, it is ultra fast. We actually changed the characteristic of two elite local varieties from normal rice to sticky rice. This is just, we did this as an example because it's important to produce varieties, for big growers, but it's also important to produce varieties for small farmers. And that was done in 2 generations that at the end of it was just 1.5 years what it took us. This is important because small farmers, they know how to grow their rice. They look at their field and say, this rice needs to be watered, or it doesn't look very healthy. It needs a bit of, nitrogen or whatever. So what we gave them was an exactly same variety. They didn't need to learn a new variety of rice, but this one produces a sticky rice. So it is their choice now what they want to grow, but they do not need to learn how to grow a new rice variety. And that was the example. It's a really nice one that you can actually take with you. It's all published. I can provide the paper whoever wants. Now, with CRISPR, what we're doing here in Australia now is we're transferring those things. We have the same genes in sorghum. So we are transferring some of the mutations that are important for rice. We are now transferring for sorghum because there is a lot of rice research, but there is not so much sorghum research. CRISPR allows us to get the knowledge from rice and put it into other crops. And that was just example, you know. And now in the last 10 minutes that I have, I'm going to tell you what is coming. All right, CRISPR at the very beginning was just about producing mutations in places, but today it has progressed quite a lot. There is a lot of development. A lot of labs are working on it. My lab is working in new technologies using CRISPR, and there are quite a lot of labs around the world doing it. Now we have not only the original SP-Cas9, but we have new Cas nucleases, and we have technologies like base editing, RNA targeting, epigenome modification, and prime editing. I'll give you a little bit of a taste of each one of them. Let's talk about the normal Cas9. This is the guide RNA that recognizes the place where it got to be. It has one requirement, and it is that this sequence needs to be followed by NGG in the genome. So the next 3 bases in this particular part of the genome need to be NGG, N being any base and G being one of things, as a normal, DNA base. All right. That is a bit of a problem because sometimes you don't have that, and you really want to cut right here. But that's not a problem anymore. There are a lot of different Cas that you can find. Other Cas9 like XCas9 now can cut in places where you have only NG or GAA or GAT. There are other Cas that have different of these bases. They're called PAMs, protospace adjacent motifs. So now you have quite a big variety of Cas proteins that you can use. You also have Cases now, that have been mutagenized, that instead of cutting the DNA, the double strand of DNA, only cut one strand. It's called a Cas nickase. You have Cas that actually doesn't cut anything. It's called dead Cas. They come into position, but they don't cut anything. You might say, well, what do I use that for? They are very useful. Believe me, they are very useful. There are Cas like Cas12a or CPF1 that have different requirement than Cas9. For example, the requirement for the PAM, the protospacer adjacent motif, instead of being on the right-hand side, is on the left-hand side of the, and is T-rich rather than NGG, G-rich, have a shorter guide RNA, and it cuts in a different way. While the normal Cas9 cuts, in flash ends, this one cuts in what we call stagger ends. Is useful. Let me tell you about base editing. Base editing, most of the QTLs that we have in crops are very simple changes. They are SNPs, they are single nucleotide polymorphisms. So it would be useful to be able to change one specific base from an A to a C, for example. But that's what base editing does. They have 2 kinds of base editing: CBEs, or cytosine base editor, that actually change a cytosine to a thymidine, or a guanosine to an adenine, and adenine-based editors that change an A for a G or a T for a C. They're called ABEs. How do they work? They use a Cas that is a nickase Cas. It doesn't cut the entire DNA, only cuts one strand of the DNA, and artificially you join an enzyme to that Cas. It's a cytidine deaminase. Now, that enzyme will take the amino group of cytidines. So when you put the Cas into position, it's gonna nick the DNA, and then it's gonna take the amino group from cytidine, and it's gonna then convert it into a uracil. And that uracil, when the cell reproduces, will end up being substituted for a thymidine. So there you go. When there was a C, now we have a T. On the other kind of base editors, it uses an adenine deaminase fused to the Cas enzyme. It does pretty much the same thing, but this time you have an adenine here, and this adenine deaminase will modify the adenine, producing an inosine, Inosine is not a natural base in the DNA, but when the cell reproduces and it copies it and replicates, that inosine will be changed into a G. So you go from an original A to a G. How can you use that? Example, very easy one. A lot of herbicide-resistant genes only have one single change that makes them herbicide-resistant. And that has been done. Here it is in watermelon. Base editing in watermelon, it changed one single base. So you change one single amino acid in your protein, and all of a sudden now you have watermelon that is resistant to herbicide. Not only watermelon, it has been done in other plants. In this case, it's in maize. You change one single base, and then your enzyme now with that amino acid substitution is now resistant to herbicides. And this is done in one single shot. Aside from that one, there are other cases that are different. They don't target DNA, they target RNA, they target single-strand RNA. This is Cas13, and it's the same thing. It's got a molecule of RNA and a protein called Cas13. It comes to a molecule of single-stranded RNA, and it cuts it. Now, why could that be useful? Well, let me tell you. Viruses, plant viruses, many plant viruses are RNA viruses. So if we were able to introduce Cas13 and a guide RNA that is targeting not a plant gene, but a viral gene, it could provide resistance. Not only that, if you could actually put several guide RNAs, you could have resistance to several viruses. Has it been done? Yes, it has been done. It has been done. Two different viruses, the rice-striped mosaic virus and the southern rice black-streaked dwarf viruses, they have been targeted with this technology, and it works. It protects rice. And you can see here, rice that is protected because they contain the Cas13 targeting the virus, while those that do not have it are not protected. Epigenome modifications, you can actually change the epigenome. Now, the thing is, in animals, the methylation in DNA is normally, as soon as the cell divides, is reset. But in plants, it's not always reset, and it actually is inheritable. So you can join Cas to a protein, you put it together in a single gene that can actually demethylate promoters to activate genes. Or you can do the opposite. You can methylate promoters to deactivate genes. Now, remember this. Here, we're not changing a single base in the genome. The genome is identical. All right? We're changing the methylation status. And this is a paper where they've done it. Look, this is a gene that when you have a mutation, you have late flowering. And this is a plant with a mutation in the gene. And these are plants. The gene is perfect, it's wild type. There is no mutation in this gene, but they have demethylated the promoter of the gene, silencing the gene, and now you have late flowering. And the latest one that just come around is prime editing. In prime editing, this is a complicated one. What you use is a guide RNA that has two different domains, all right? At one end of it, you have the normal one, that is the one that you use for targeting. And on the other end of it, you artificially add something that contains a little bit of sequence and the modification that you want to produce. Now, the trick here is that you join Cas9 with a reverse transcriptase. So when it comes into position, this Cas9 is a nickase. It's going to nick one strand of the DNA. This is the part that recognizes the place where it has to go. And this is the one that you're going to use as a template. The green part and red part is going to be used as a template. This reverse transcriptase that is here will get this template and copy it in reverse transcription. After that, you're going to have one branch that is a bit longer than the other, and then it's called flap equilibration. The cell will repair itself. It will incorporate what you wanted that was here. You actually program what you want to put. And after replication, you have your edited DNA, meaning you have exactly what you wanted, where you wanted. This has been done too. You can see it here. This is the sequencing. The wild type got TGG. This edited plant got TGC. And that actually confers these plants with herbicide resistance. But when you say herbicide resistant, you could use it for anything at all. Herbicide resistant is very easy because you put the plants with herbicides and those that are wild type die. Those that have been edited don't die. So to finish up, the challenges, what are the challenges? Well, look, this is a very new technology. Everybody's talking about it, but it's really, really new, okay? So the delivery method, you know, some elite varieties in some crops are very difficult to transform. So we need some variety independent platforms to be able to gene edit them. Some of the methods are not very efficient yet. I didn't talk about gene replacement, but you can actually do gene replacement. You can actually change an entire gene for a different version of the same gene. That is still not very, efficient. And of course, it's the government regulation. And let me remind you, random mutagenesis, where you produce 50,000 random mutations, is not regulated. And we have 2,000, more than 2,000 of them being grown in Asia. While genome editing that produces one single mutation, extremely precise, will it be regulated? But I'm happy to see that Japan has taken the scientific route, you know, Australia too. SDN1 means when you do normal genome editing, is not regulated in Australia. And with that, I finish. I, of course, need to thank all the collaborators that I have standing, you know, in the Chinese Academy of Science, here at the University of Queensland, different universities in China, the US, and Pennsylvania. Of course, I'm always open. If anyone wants, you know, to explore any kind of collaborations, we will be happy to provide assistance or any kind of collaboration, any kind of help we can for anyone who wants to get started into genome editing. Thank you very much.
Okay, thank you, Jimmy. So it's a very clear and very useful talk. So especially with your images, so several images, I would like to use in my lecture. Okay, it's very easy to understand. Thank you. So now I got some question from the chat. So it's very simple, but very basic question. How can poor countries benefit from these technologies? Is there any other ethical concerns of this technology? So this is not so directly related to your presentation, but it's very important question, I think. So if possible, please answer the questions. You mute. You leave the mute.
Sorry.
Okay.
Poor countries are actually the ones that can benefit most out of this. My example gave you a good example of, you know, small farmers. Gene editing is so easy that we can actually, rather than get big multinational that grow humongous farms, you know, we can actually get varieties improved for small farmers using gene editing.
Mm-hmm. Maybe now that we can say it like that, yes. So in general, so the small companies have the benefit to improve their own cultivars in the vegetable and some things as the crops, I think so. So, and another question, does gene edit plants need to follow the GMO regulations? That means in Australia, in New Zealand, and it's a very delicate problem. I think I don't know where. So, if possible, so GMO regulation in Australia, yeah.
Gene edited plants, as long as you don't provide with a template, they are non-regulated in Australia.
So
we do have plants already in the field. We got plants in the field, in field trials. As long as we can provide proof that they don't have the transgenes, they are not regulated. They're called SDN1 type.
Yeah. So maybe I think it's the same as Japan. So the STN1 is non-regulated GMO, but STN2 is great, I think. Yeah, STN3 is completely GMO, right?
Same in Australia, yes.
Yeah. Okay. Yeah, and almost of the chats. So thank you for GB. Yeah, so that's been no questions. So, so this is very good question. So what are the implications of gene-edited crops on overall agrobiodiversity? So that means what's the role of the gene-editing techniques for diverse of the agrobiodiversity?
Well, let me explain one thing. We can actually create a lot more diversity with gene editing than we can with anything else.
Yeah.
That's one of the things. That is one of the things we're doing. We're actually artificially creating variation in genes to get alleles of the gene that are not present in natural population. You can actually increase it. But let me explain this. Agriculture is completely opposite to genetic diversity. Agriculture is a very artificial thing that humans do, and what they want to have is just the opposite of genetic diversity. They want to have homogeneous plants.
Yeah, I see. Yeah. Okay. Thank you. So another question is gene editing expensive? Is the technology patent? That means patent about about patents. So Professor Ezra already answered. So that is gene editing expensive or not?
Okay, I'm not going to talk about commercialization. For that, you will need to have a license to exploit it, okay, for commercialization. Now, gene editing is so cheap, you wouldn't believe it, okay? Let me explain this. Before CRISPR, gene editing, when you wanted to create a gene editing by zinc finger nucleases, for example, you needed a super specialized lab. You needed a few months to clone, the two constructs that you need, the molecular biology to do that. In my lab now, we can prepare 5-10 constructs in 2 days.
Okay, thank you. So that's just only the last question. So please comment on off-target activity of these techniques.
Good question. We have done in several, at the beginning when we started doing gene editing, we used to sequence the entire genome of some of our plants, and we found that pretty much it was single target. We did not find a single mutation anywhere else in the genome. Now, let me explain this. Specificity is very important. Off-target is very important for humans. Like, if you all of a sudden you're trying to cure a genetic disease in humans and you affect other genes, and then all of a sudden, you cure one disease, but the human, start growing 10 fingers in each hand, that's a problem. But for plants, it's not a problem because we can produce 100 lines. Nobody cares if it's got an extra mutation because we throw that away. But the truth is that it's extremely precise.
Okay, thank you. So more than 50 questions I have, so it's impossible to pick the whole. So I'm very, very sorry. So that means the time is a little bit overdue due to the incompetence of me. So I'm sorry. So okay. So thank you, Professor Jimmy. Okay. Thank you very much for your good presentation. Okay, so next time is for poll questions. I pass please, Dr. Bibha, please. Thank you, Jimmy.
Thank you so much. As the organizers, APARI, KPCH, and BCIL, we want to have a small survey conducted during the webinars. So we will be posting 2 questions just now and request all the participants to kindly click the options. As we all heard, that gene editing is opening up new approaches for crop improvement. Where do you think it is most effectively employed? There are 4 options: improving resistance to the disease and pests, better nutrition or functional qualities, improved climate change resilience, all the above. So may I request the participants for another 1 minute, this will be on the screen, to kindly click on the option that they think
which of the following statements are applicable to gene editing? Gene editing is a precise breeding tool. Gene editing can be used for deletion, replacement.
I'm sorry that somebody please mute. Keep the mute.
Gene edited products are indistinguishable from products of conventional breeding.
I'll read the second question. Gene editing is a novel approach. We all heard just now that there are continuous advancements being made, as was very nicely explained by Dr. Jimmy. Which of the following statements are applicable to gene editing? It is a precise breeding tool. It can be used for deletion, replacement, or insertion of gene sequences. SDN1 and SDN2 gene edited products are indistinguishable from the products of conventional breeding, or all the above. So request the audience to kindly list the option and submit. Give another 30 minutes. We shall be sharing this type of a small poll will be conducted throughout the 3 webinars, and we shall be sharing the results with all of you at the completion. Thank you so much. I think, Dilip, it would have been done. Yeah. Thank you so much for your participation. I will give it back to Professor Osawa to kindly lead the panel discussion. We have very distinguished panelists here, as was already introduced by Professor Osawa. So
Okay.
Proceed, sir. Thank you so much.
Okay, thank you. So after that, you should display the result of this poll, okay?
Thanks, sir. We will do
Okay.
that.
Okay, so the next one, so in the panel discussion, we progressed in the panel discussion. We have experts from the 5 countries in the Asia Pacific region. So from the Bangladesh, India, South Korea, Taiwan, and Malaysia. The panelists are, so I introduced in each person. So Dr. Zeba Islam Siraj, Professor, Department of Biochemistry and Molecular Biology, University, Dhaka, Bangladesh. Shook hand, okay? Yeah, no problem. So the second, Dr. Sharma, Deputy Director General of the Crop Science, India Council of Agricultural Research, India. The third, Dok Han Kim, Vice President, Future Food Resource Forum, Korea. Dr. Kok Gan Chang, Deputy Head of Department, Institute of Biological Sciences, Faculty of Science, University of Malaysia. The last person is Chuan Yang Hong. Sorry, it's very difficult for me. So the Professor, Department of the Agriculture Chemistry, National Taiwan University. The panel discussion will focus on status of research initiative, the potential challenge related to gene editing in agriculture in Asia, in your country. I request each presenter to briefly inform about the status of research and potential in their respective countries. So I'm very sorry, the time is very consumed now. So each panelist may share their views in almost about 5 minutes. I will start alphabetical with the Bangladesh. So the Dr. Zeba, please.
So I won't show any slides because it's the time is short. So thank you very much for a nice introduction, and thanks to the organizers for including me as a panelist. So in Bangladesh, CRISPR is being done in a few laboratories only. So for practical purposes, the CRISPR is being designed against wheat blast. So this is in collaboration with some labs in the United Kingdom. And the wheat blast was quite a big problem. So they are using CRISPR to downregulate susceptibility genes, which were identified before. So that's in progress. And in my lab, I'm working on salt tolerance. So we identified certain genes which were upregulated in traditional salt-tolerant land races. So we, sorry, they are downregulated. So what we did is we took a commercial variety and then we downregulated those genes and we got salt tolerance. So they are mainly transporter genes such as the AKT1 gene. We downregulated it and got tolerance in a commercial variety. And the other one is a ABC transporter, which we downregulated and we got salt tolerance. So otherwise, there are many problems in Bangladesh. So we heard this morning that, you can increase rice yields. Bangladesh, the main problem with Bangladesh is we have, very low amount of land available. It's a small country and we use mostly rice is grown. And we actually have, for many areas, we have 2 cropping systems of rice. And we also need to enhance nutritional quality because, particularly as the soil is being depleted with the, the 2 cropping system of rice. So we need higher yields for most crops. And then, of course, there's the pest and the pest resistance and the disease resistance. So, and we heard this morning that if you use Cas13a, you can directly, target viruses. So viruses is also a problem for many crops in Bangladesh. So CRISPR has a really wide applicability for, and of course, the main problem is, of course, abiotic stress tolerance because the salinity is increasing in the south, due to climate change. And in the north, there's the drought is increasing. And, we have examples where drought tolerance was increased. I think it was in America. They downregulated a certain gene to get drought resistance. So as I was saying, there is a lot of potential in Bangladesh, but there are only a few labs currently working on CRISPR. And, as Dr. Jimmy said, Botella, that it's really quite easy. And, master's students have been doing the, the CRISPR cloning. It's not that difficult. So I can, from my experience, I can, confirm that we are doing it and it's not that difficult or not that expensive. So, and we use actually, there were some questions which weren't answered, due to the lack of time. So we use actually for rice, I use in-planter transformation, which is we don't go through tissue culture. So it's actually quite easy and it's genotype independent. And then of course, you have to select for the non-transformed plant if you want a non-GMO. So you can do that, and maybe we haven't done that yet, but we are, hopefully we can do that if the plants are quite tolerant. So we are now working at T2. So that's about what I wanted to say. Thank you very much.
Thank you. Thank you very much. So that means, are you close to the breeding ability in Bangladesh? Not yet.
Sorry?
Yeah, so it's close to the make a breed the new varieties using gene editing in Bangladesh. Not
yet.
Not yet, yeah. A little close or not so far?
No,
yeah, okay, thank you. Thank you very much. So it's a very big problem for the climate change and so the sanitary resistance. Okay, I think I can understand. So the next one, so the Sharma, please. Zeba, thank you very much. Okay, next speaker, the Sharma.
Yeah, thank you very much, Chair. Good morning, good afternoon, and good evening to all. I am from Indian Council of Agriculture Research, Government of India, this organization is more than 93 years old, and we work on more than 85 crops, field crops, and 145 horticultural crops. You know that in India, we adopted these new plant breeding technology very fast, particularly in cotton. Transgenic is one of the major example of India. And then subsequent to that, we are working on genome sequencing of crop plants, genomic assisted breeding, marker assisted selection, and now coming to new plant breeding technology like genome editing. So here I would like to tell you that not only about the basics, which Dr. Botella has covered very nicely, the basics are related to genome editing. And I would like to give you an idea and small picture of Indian initiative in genome editing. So we have basically in crop plants two funding agencies. Particularly, there is a program on genome engineering technology and their applications for human resource development in cutting edge genome editing technologies, Indo-US Genome Engineering and Technology by the Department of Biotechnology, and then individual-centric and multi-institutional R&D projects are being funded by Department of Biotechnology, Government of India. Indian Council of Agriculture Research also fund network projects. I will tell you in a few minutes that what we are doing here, but National Agricultural Science Fund of Indian Council of Agricultural Research also fund network projects on genome editing. And if I tell you in brief, Department of Biotechnology have funded more than 24 projects with the cost of around 2.1 million US dollars. And similarly, ICAR, Indian Council of Agriculture Research, has funded 4 projects with a budget of around 1 million US dollars. And Department of Science and Technology, Government of India is also funding some projects. The number of projects which we have total is now till date 32 in working on different crops, and total budget which Government of India is spending for the past 3-4 years is around 4.2 million US dollars. If we see a number of plant species which we work on transgenic crops, and then as I told you, 32 projects, and then more than 41 scientists are engaged in doing this type of work. If we see the crop-wise distribution, as told earlier, rice is one of the most important crop, and in India, more than 50% of the project's budget is going to the rice itself, and then followed by a small budget to different crops. And I will not go into the details of that crops, but under National Agriculture Science Fund, genome editing in rice for enhancing stress tolerance was done at our institute, Indian Agriculture Research Institute, where we did loss of function mutant of drought and salt tolerance, DST gene, encoding zinc finger transcription factor, which confers drought and salt tolerance in rice. And all these plants are now, we harvested seed and are now in second generation of its growth. Similarly, these DST mutations conferred enhanced tolerance to drought, and which has been now recorded by using automated phenotyping facility. And we recorded photosynthesis and thermal images of 85-day-old plants, and we found that the genome edited plants are really superior to that of their wild type. Similarly, loss of function mutation of fine culm1, FC1, teosinte branched 1, TB1 gene increases tillering in rice. And that work was done under the funding of National Agricultural Science Fund by International Center for Genetic Engineering and Biotechnology, New Delhi. And we edited one of the most important varieties of rice, MTU 1010, for this TB1 genes, and there was more than 60 to 70, 66% to 70% higher increase in the yield of these edited plants, which we observed in laboratory stage. Similarly, dense and erect panicle in rice, DAPI1 genes for G protein alpha subunits involved in the regulation of number of grains per panicles. And by editing this particular gene, we could get more than 54% higher grain number in genome edited plants. In case of rice, again, we are dealing with change in architectures and natural mutation in ideal plant architecture, that is IPA1 gene, which prevents miRNA 156 mediated degradation of IPA1, increases grain yield in rice. And we showed very clearly in case of Swarna variety of rice that there enhances the yield potential of this particular variety. Similarly, we are collaborating with other countries, and we recently signed an MOU from ICAR side with Heinrich Heine University of Germany for procuring genome edited rice lines which are resistant to bacterial blight of rice, wherein 3 genes for bacterial blight resistance, these sweet 11, sweet 13, and sweet 14 gene were edited. And one of the most important examples which I would like to quote here in the Department of Biotechnology Institute, National Agri Biotechnology Institute, Mohali, there we edited lycopene epsilon cyclase LCY mutant genes in case of banana, and we found that it increases the carotenoid content extensively in edited plants, and these plants are now at various stages of development, and these have already been what you call grown in the net houses. So as far as regulation of genome edited crops are concerned, we are very well aware of in discussion stage, and it is being the regulations are being formulated by the Department of Biotechnology, Government of India, and we have designed a very particular
Slide share, sir. Slide share.
I don't have slides, so I'm just telling you without slides. That SDN1 and SDN2 would be considered similar to that of natural mutants, whereas SDN3 would be considered as a transgenic plant, and that's what we have now recommended to the regulatory agencies, and I'm sure that it will be very soon cleared by this agency, and we have our plants in the field as well as in net house testing, and those are being now evaluated for their different traits. And so we have seen very nice example from Japan now for GABA gene, which was presented by one of our first speakers. And similarly, there are many examples from the world, like US has approved Vaxicorn, US has also approved CRISPR-Cas edited mushrooms, and also soybean with high oleic acids. So this particular technology is attracting a lot of attention world over, and But still, I think the draft guidelines for genome edited plant across the countries are being discussed, and we have to be very clear what we have to consider it as a natural mutations, particularly SDN1 and SDN2. Number two, the way forward, which I would like to tell, is that a mission mode program on genome editing of crops for food and nutritional security and climate resilience, which we are starting, and we are investing more than $2 million US in Indian Council of Agriculture research in coming 5 years, and also at selected crop species and crop plants for a specific traits, the traits which are controlled by single gene and particularly the down-regulated genes, and which can be manipulated by single gene mutations by using all those techniques which are explained by Dr. Jimmy Botella. And all these techniques are being covered by all the labs in our 26 labs of India. And I'm sure that with the passage of time, we are going to invest more in this particular new plant breeding technology for the benefit of farming community. So with these words, now I close here, and I welcome all the questions from the participants. Thank you very much, Chair.
Okay, thank you. Thank you, Dr. Sharma. So maybe the first your presentation, so we can't use the GE until the analysis of the traits progresses is needed. So after that, you will try to the gene editing techniques in India. Okay, thank you. After that, we will discuss about your country's situation. Thank you very much. So the next one is I'm sorry, I'm waiting for you. Sorry, Dr. Kim, please.
Okay, thank you. Can you hear me?
Yes.
Okay. Thank you for. I'm very delighted to be here. Such an important webinar series for sharing the information about the GE research initiative and seeking the possibility to get the harmonized regulatory landscape. And I was asked to introduce Korean's research status about this GE crop development, and I will give you a briefing as simple as possible. Actually, the Rural Development Administration, which is the government agency responsible for the agricultural biotechnology for crops and livestock in Korea. Okay, in this century, the RDA has invested for 20 years to develop the agricultural biotechnology like GMO and LMO. And then I witnessed that the great improvement in research capacity in Korea. And recently the RDA decided to invest in genome editing, and then decide to install the start the national programs, and then install launching a new breeding technology center to support the R&D activity in GE research and development. And the size of the funding for this center is about the 65 million to 70 million US dollars, and the duration is about 7 years. And about 90 principal investigators or academic professors participating in this program, and then they are doing their own research R&D projects. And the projects are divided in two groups. First one is the basic science or the platform development research, which is about developing new genome editing technology, and then phenotype analysis for core collections to find useful traits in the core collection. And the second part is the application of the technology to actually breed useful cultivars and varieties. For 20 years, when the BioGreen 21 or next generation BioGreen 21 program was going on, we have focused largely on rice, because as you already know that the rice is very important in Asia area. But in this New Breeding Technology Center program, we slightly change our focusing area from rice to other crops such as cash crops. The research purpose or objectives of these projects are first one is changing or replacing GMO with these GE crops. So by introducing herbicide resistance or pest tolerance. And as also we are doing some research projects to increase the biotic and abiotic stress tolerance. And we are doing also the research projects the developmental improvement, like the plant shapes or the viviparous premature germination, germinated thing. And then we are also doing the research to improve nutritional or functional value of the crops. In brassicas, there are a variety of functional substances which are good for human health. So the researchers try to improve those or increase these nutritional or functional substances in this crops species. We are also doing the research to add some functional oils in soybeans, and we are also doing some researches to increase carotenoid in pepper and carrots. In Korea, we have some companies who are doing this research and development for their own crop breeding. And so about 6 to 10 companies, private companies, are involved in these projects. And then about 20 to 25 universities are also participating in these projects. So I think the genome editing is very important for Korean agricultural biotechnology, because there is a huge number, not huge, but substantial numbers of people who are against GMO or genetic modification technology. And we believe that the GE will give us some chance to overcome these kinds of hurdles, so that we can contribute to our agriculture or global agriculture by developing these GE crops without any problem from the public negative perception. Thank you very much.
Thank you very much for your brief presentation about the status in the Korea. So I know, wow, the very difficult situation in Korea, so something, okay? So thank you very much. Okay, the next panelist is Cham, Dr. Cham, please.
Thank you, Professor Oshawa. Good afternoon and good morning to everyone.
So my name is Kokan. I'm from the University of Malaya, Malaysia. And a brief introduction about the plant biotech research or the crops biotech research in our country. We, at least for our department, we have the research on the CRISPR-Cas9, and I know that my colleagues are actually working on RNAi interference on the flowering flower, and also high grain size, high production and the grain size and pest resistance of the rice using the what we call gene editing techniques. And also we have a transcriptome research on the flowering in our country. And myself is working on the CRISPR-Cas9, but not on the crop biotech, but on the bacterial biotechnology. And also I'm working with a colleague who has retired on the crop sensing enhanced plantlet transformation yield. So, and we also work on our department also work on the drought resistance using the gene editing techniques in the country. Of course, being one of the largest palm oil research. So there is a lot of research working on these, what we call cash crops, as well as other major commodity crops in our country. And at the moment, I think the plant biotech research, including the crops research, are quite active in Malaysia. Although I mentioned some techniques that has been used in our country. And I would like to also draw to the attentions of the audience that of late I have read about latest techniques in gene editing, which is the Retron recombinering that has been reported in PNAS by the Harvard group, McDonald Church. I think this is another thing that could be very interesting. I share with some of the concerns that after Jimmy has presented his lectures, and some audience put in the chat box that how can we distinguish the GM plant using these very novel gene editing techniques, which I agree being a regulator in the country. So CRISPR will be posing some major challenge in the regulation. So perhaps the Retron could be a better way for the regulator. At least there is what we call a signature sequence that we can actually tag to the offspring of the genetic engineer crop. So thank you, Chair, for this section. Thank you very much. That's all from me. Thank you.
Okay, thank you. Thank you very much. So, so I'm sorry, so the Mr. and Mr. Alia, so you try to share your
yes.
So please stop.
The message. Please, somebody is trying to share the screen. Kindly unshare the same, please. The Secretariat is also trying to stop it, sir. Just give us a few minutes. I think we can proceed with the discussion. These people will remove, sir.
Okay, so okay, so this bill is a little bit curious for me, so but we continue the last one. Dr. Hong, please.
Thank you.
Yeah.
Hello, I'm Chan Yang Hong from National Taiwan University in Taipei. In my lab, we are focused on the generation of crop with stress tolerance and with low heavy metal. But today I want to share our research with you. Today I'd like to share the current state of our government's genome editing policy in Taiwan. And in Taiwan, high humidity and temperature severely affect crop pests and disease susceptibility, which in turn affect crop health and our production. And fortunately, more frequent than the more intense climate events, such as drought and rainstorm, have both been observed in recent years. Since last year, we have suffered from the lowest drought in more than half a century. And we know typhoon is the major source of water in Taiwan. However, no typhoons made landfall in Taiwan last year. It's the first time since 1964. And consequently, most of islands' lake and reservoirs have nearly run dry. and water restrictions have forced many residents to modify how they shower, wash dishes, and flush. And just a couple of weeks ago, things changed dramatically because rains hit South Taiwan. You may know that there's another typhoon approaching Taiwan in the next 2 days. So to take action to combat the climate change and its impacts, our government encourages researchers to carry out gene editing research. And in recent years, we have invested in gene editing research and development on staple food crops, fruits, vegetables, and flowers, and hope to improve their resilience to climate change by SDM1 type gene editing. We know many countries have exempt SDM1 type gene editing from GMO regulation of crop plants. So although the legal status of genome editing is still under construction in our country, we continue to communicate with stakeholders and collect relevant opinions. Our country is still in discussion on how would we manage gene editing products. We would carefully regard the international trend on the regulatory and the management issues on this precision breeding technology to closely in line with international consensus. Thank you very much.
Sorry, thank you very much, Dr. Hong. So that in your country also it's a problem. So the regulation of the GE or the GM, right?
Yes.
Yeah. Okay. So today, so it's maybe all panelists. Thank you very much for introduction in your situation, in your country's situation. So I know well. So almost of them. So many countries have the. It seems to be difficult to distinguish between. Wow. Okay. So the GM and G modified plant and G editing plant in many countries, I think. So it's a big problem. But to maybe how to do that, how to think about that. So the webinar third, it's we have the chance to discuss about that. So in this panel, so I'm apologize for my inattention, and the scheduled time has a little bit passed. So, but we try to discuss among the panelists and also the presenters. Okay, so maybe from a technical point of view, I think the Jimmy's lecture was very quite positive to the Asian countries. How are the patents for all the technologies? I asked to the Jimmy. So you present the post CRISPR-Cas9. So you presented that. So the almost technique is show the patent needed to get the patent from each company or each what I should say. But please, if you have any information about the patents, the new techniques. Jimmy, you have?
No, sorry, I apologize. I'm not into, I don't know the IP position for the new ones, but most of them will be from Jennifer Doudna, whose group has been the one developing most of these new things. If you have noticed now, every single paper that comes out with new CRISPR advances, it includes a little bit about plants. Before it was only about humans. Now every single paper with any like prime editing came with an example in plants. Yeah. The reason is because you need that example to put it on the patent.
Okay. Thank you. So the old panelists say so under the developing and the techniques using for improving the crops. So under the study, it's no problem. But if they try to use to improve the crops, so all new techniques also regulated by the patent. What do we think about that? So Professor Hiroshi, you have any idea, any information that?
Okay, so I think that all patent holder of the company is they have their own policy how to license, and but I think that in general even a CRISPR patent, if the objective to use this new technology is for supporting a developing country, that will be maybe some license will be very easier, but only the objective is for commercial use. That's we need to make a huge discussion. And actually, like Corteva say that if they wanted to, if this technology used for the supporting a developing country, improving some staple crops like cassava, they can support to use this techniques. So that's why it's depending on the occasion of how to use this technology. Yeah, so but some country like Japan and Korea and some other countries, maybe we have to get patent. But yeah, but discussion is can open the how to use the technologies. Yeah.
Thank you. So maybe the understanding the no problem. So in Japan also the discussion, the patent is will regulate the improving the cultivars. But so the experience of the Hiroshi, so not so problem. It can be cleared, create this patent problem. So about that problem, so every panelist can have any question to Hiroshi or Jimmy.
Sir, I just wanted to intervene and say that on the IPR issues, which is the patent related issues, we have scheduled a presentation, a detailed presentation on the topic in the webinar 3. So we will have a lot of, interventions that can be, answered or clarified here also. Yes, sir.
Okay, thank you. So
if you permit, shall I show the, if you permit, then shall I show you the results of the poll, sir? We have already got the result of the poll.
Okay, sorry. So that's bad. Everybody very interested in the patents. Okay, so my question is the first. Okay, so the another question. So maybe for example, the Dr. Zeba asked presented the traits. For example, the salinity tolerance and abiotic tolerance, the biotic tolerance. So how to use the gene editing techniques to improve the such a tolerant, abiotic and the biotic tolerance? To ask to Jimmy and Hiroshi, and also if possible, please.
The main problem about improving biotic and abiotic stress in crops is to know the biology of it. So what we need is a lot more knowledge in the fundamental basic biology before we can actually target genes. There are some genes that we can already target for biotic stress and abiotic stress too. But our main gene editing is not a problem there. We can do gene editing, not a problem. What we're lacking is enough biological information about the genes that can actually improve those crops. That's why we need a lot of geneticists, a lot of physiologists working on this kind of problems so they can identify the targets.
Yeah, I think so. Yeah, so the first we need to get the information about the genes related to the traits. Okay, so I think that Hiroshi have any idea to
the question. As everybody knows, there is a many report describing the biotic resistance genes. But the important point is to maybe to prepare a publication that extreme mutation is very important for preparation of the understanding of the function of gene. But for the crop breed, maybe we need different alleles to show a more moderate phenotype which has some effect on the other developmental pathways. So so far we have many ideas for target genes, but those genes is for used for publication. But for the actual application, to create allele in the target genes. So that will be more.
Thank you. So the please mute. A little bit noisy, so I cannot hear the discussion. Maybe okay. Thank you. Thank you. I'm sorry, Hiroshi. So you are with answer.
We understand sometimes.
Okay. So among the panelists, do you have any questions? Everything. So among them. So for example, from the Korea to Bangladesh and something, Korea to Taiwan, and do you have any question or suggestion, something?
Dr. Osawa, yeah,
okay, thank you.
Actually, the actual research and development for this genome editing is a matter of science and business.
Yeah.
So the technology should be improved and then developed further to contribute to this Asian agriculture.
Yeah.
But the most important thing is the regulatory harmonization, I think. And the main purpose of this webinar series is to seek the possibility to get such harmonization. We have observed that we made some mistakes at the beginning of this genome era, so that there are huge anti-GMO activities from anti-biotech groups, and then as a result, many countries or many regions or many governments regulated these GM crops very strictly, so that the potential of this fantastic technology went down, and there. I think that we should not do this mistake again, and we scientists and then industries, research institutes should come together and then do something together to make sure that we can do without any problem or without any barrier.
Yeah.
So I think that is the key point what this webinar series for.
Yeah.
Thank you very much.
Thank you. Thank you. So it's concerned about that. So Dr. Hong from Taiwan, do you have any ideas or any question or something? Okay.
Okay. Ryo, can I have some comment on that?
Okay. Please, Hiroshi. Okay.
Okay. Thank you very much,
Kim.
Dr. Kim, for the good position. So in Japan, during the development, we have a very good harmonization with the government and academia, and also the companies. So those are the different stakeholders make a big discussion to how to get acceptance of these new products. And then maybe one extreme issue in Japan is in Japan there is a many scientific society of plant breeding or plant horticulture, and in Japan those scientific society can made a statement about this new technology by year by year. So then those are opinion from the scientific community is quite supportive to proceed our products to the society. So that's but I discussed with some other countries, but such activity is not so strong by like academic support from for the developers. So that is one point which we are hopefully successful in Japan for the commercialization of new products. That is one point of view of my Japan.
Yeah, thank you, Dr. Ezra. And I'm totally with you on your point. So thank you.
Yeah. So it's a very good point. So the president, so the Hiroshi is also the president of the biotechnology. So and I supported this GE technique for the improving the crops by the plant breeding society, the plant breeding. It's very important thing. So what do you think about Dr. Hong?
Can I ask a question to Dr. Ezra?
Yeah.
I'm curious about the is that expensive to get a license from the three company?
Okay, that's a good question, and as already patent holder, they notify that it's depending on who is going to commercialize this patent, and so we get the license as a venture company from the university. It's a very small company, so patent is reasonable, no? So that's why we could commercialize it. It's depending on the companies and the size of the company and the business of the company. That is the same with other patent, not only this CRISPR patent. If we use another patent, the patent license will be changed depending on the situation of the who wanted to have this patent. So, but in our case, in Japan, yeah, it was reasonable.
Professor Rai.
Thank you.
This is Dr.
Shama, okay, please.
I have one or two questions to Professor Ezra about his products, which he has developed, particularly the amino acids GABA, which he has produced in tomato. So, have you, Professor Ezra, how much quantity do you produce in say single tomato of 100 gram size, and how much you recommend that one should eat, because it is a neurotransmitter, and this particular amino acid is recommended for many neurobiologists for certain ailments related to headache, migraine, and all those stuff. So how much one should eat?
Okay, thank you for the question. Can you see that tomato photo behind me?
Yeah, I can see.
Just one fruit is enough. The content of the GABA in just one fruit is enough to get the expected functions. Just one fruit. It's a 20 grams of fruits is enough to get the expected function. So it's easy to take. Just one fruit, small fruits. Yeah, you can have every day.
What is the recommended dose?
Recommended dose is 15 milligrams.
Okay.
The one fruit contains 15 milligrams. So that's why I say that the one fruit is enough to get the expected function.
Professor Rai, I would like to add a few more comments from my side, particularly regarding crop improvement. Most of the programs are now concentrated on those crop species where genes are well characterized, number one. Number two, where genome sequence is available in public domain. Number three, where transformation protocols are available. So these three prerequisites, if in any crop plant, if these are available, then these are being considered for genome editing. But we have many horticultural crops where nutritional improvement and resistance to biotic stresses is very, very important. And in those horticultural crops, we don't have even transformation protocols available with us. Most of the genes are not well characterized as we have in rice, in tomato, in Rhabdopsis, and other model species. So what is your opinion about those horticultural crops?
Okay, thank you for the question. And I think it's depending on the trade and in a horticultural crop. And if we increase the GABA content, and the mechanism of GABA accumulation is in the same in the all horticultural crop. And then if we can use, that means that we can use the same strategy to increase the GABA content. And also some other trade like extending shelf life. The plant hormone ethylene are responsible for the shelf life, and the crops, horticultural crop, belong to the ethylene sensitive crops. That's we can extend the shelf life by same technologies. It's depending on the trade.
Okay. Okay. So it's maybe the discussion is a heat up, so it's a very nice situation, I think. But I apologize for the inconvenience but we are at the end of the meeting time. Sorry. So the finally, I asked the two questions in each presenter. The first is asked to Hiroshi. So you are the specialist is the vegetable improving breeding. So, but I ask you, the agriculture, particularly the Asia Pacific region, is becoming more and more challenging, particularly the concern of climate change. Okay.
Yeah.
So that there is a need for novel strategy to deal with the same. So that the question is related to, for example, the Dr. Zeba. So where do you see the role of the gene editing tools in this scenario to improve crop production in Asia and Pacific? It's a big problem. You are not the president of the Asia. I know well. So, but if possible, please answer. Where do you see the role of the gene editing tools?
Okay, thank you for the question. Actually, the greenhouse effect, global warming is a very important target for, especially in Asian countries. So then I have a lot of many students from Asian country, and they are very interested in the increasing the heat tolerance of the crops. And so now my laboratory is trying to already identify the mutants showing a heat tolerance in tomato, and then now trying to we're very closing to the genes which can increase the heat tolerance. And once we obtain the genes and which has, the mutation is not so complicated, just a small mutation, then maybe we can create a heat tolerant tomato using the gene editing technologies. And also maybe the gene is the same genes with other horticulture crop species. Maybe we can use information for the improvement of increase the heat tolerance in other crop species.
Okay, thank you. So that means that the basically the same as the vegetable crops and crops. So the heat tolerance is needed, of course. Okay.
Yeah.
I think
it's a big target.
Okay, thank you. So the final important question. So I asked to Jimmy. So there is the growing opinion on the genome editing that results in the introduction simple mutations with small insertion presents profiles similar to products of the conventional breeding. What are your views on this one? So that means every panelist I also asked to this is the genome editing is GM or not. So in each country, not yet discussed about that. Maybe if you have any idea, so this question.
Well, that's an important question. Okay, yeah. That's why I started my talk by explaining about mutation breeding, because mutation breeding has been done since the 1930s, and I actually did the purpose. And that table that I put in there, I took it last week from the official archives. 2,000 different random mutagenesis plant commercial varieties in Asia. So those ones started by producing 10 to 20 to 30,000 random mutations, while CRISPR only produced a single one. That's why I always start my talk by explaining this is what we are eating today. This is a different way of doing it that is a lot more precise. Now it's a government thing, whether the governments want to approve it or not. But look, I saw a picture once from a demonstration. I think it was in South Africa. There was a lot of people there, and they have placards because it was a demonstration because the food prices have gone up, all right? And one of the placards say, when people go hungry, they will start eating the politicians.
Yeah.
So I think that is a good message.
Yeah, thank you. Okay, I think so that it's a big problem. So we plant breeders should work for such a people. Okay, so there's a big problem or not big problem. So GM or not GM. So we should use the useful techniques, useful tools for improving the crops, I think. Okay, thank you. So I'm very sorry. So more than 30 minutes over the time of this meeting. So I would like to express my sincere gratitude to Dr. Ezra, the Hiroshi Ezra, Dr. Jimmy Botella, and 5 panelists. I'm very sorry. I cannot have much more status from
But it gives 24 sound. It will give?
Yeah. Okay. Who bother me? Okay.
Have 21.
Okay, so the panelists for providing us the meaningful information in the discussion today. So I believe that today's discussion has brought the hope germinating breeding can be developed, deployed in Asia and the Pacific countries. So, and to contribute to significant to sustainable.
No, don't write anything.
Okay. 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 Please, I want to say much more. Okay. Okay. So they contribute significantly to sustainable agriculture productions. Okay. The closing remarks. So I pass the microphone to Dr. Rabi, right? No need.
Thank you. Yeah. So thank you, Dr. Osawa.
Yeah.
I think we are running over time. So maybe what we will do is I will quickly show you the poll results, which you had requested that they should be displayed. So Dilip, can you quickly show the results? Can you put the screen, please? Yes. So, sir, what all this has been a very interesting deliberation, and what we can see, the results also are, depicting the same observation that you just now concluded, that gene editing in agriculture is definitely very important, and it is opening up new opportunities. And almost 81% of the participants have indicated that this can be very effectively employed, and more so in our Asia Pacific region. Can you move it up, please, for the second question? Philip, can you move it up, please?
Okay, so that maybe these answers, this result is very in general, I think so.
Dilip, can you please move the slide up?
No need. Okay.
Visual, ma'am.
Question. Yes, please.
Ma'am, you can do it by yourself.
Okay.
Okay, thank you. So if you see the second question here again, we had asked that which of the following statements are applicable.
So you already know
indicated that all the three applications, like the gene editing being a very breeding tool,
and short for the sound, we call it as a fuel.
Please mute this part.
Please
Long vowel sound and short vowel.
Please mute.
Okay. Again,
Mute it.
Yes.
Okay, thank you.
Gene editing can be used for deletion, replacement, insertion, and SDN1, 2 are indistinguishable from products of conventional breeding. So both the questions reiterate what has been, we've had a wonderful discussions, and what has been, what has been coming out of it. And I think there are 2 points from my side. I'll quickly say regulations and the patents. This came as the, key points. And I just want to share that on both these points, we are going to have extensive deliberations in the next two webinars. And over to Dr. Ravi, sir, if you would like to say something.
Thank you, Dr. Vibha, and thank you, Dr. Osawa, for such a nice moderation. Thank you to all the speakers, to all the panelists. I will not take much time. We are already shooting above, and I will just like to say that we will be sharing the outcome of the webinar with all of you. And if you have any further more comments or questions which are not answered because of the large number of participation, please write to us. We will get back to you. We want to make sure that we really mean your input. We want a comprehensive output of the webinar, which will feed into our resource document. Thank you one and all, and thank you, Dr. Vibha, for really managing the show. And I would request Dr. Rishi Tyagi, our coordinator from EPCOP, to provide a formal vote of thanks. Dr. Rishi.
Thank you, Dr. Ravi. I'm sure that everybody has enjoyed this deliberation on this important topic. And very briefly, I propose the vote of thanks to first of all, the chair, Dr. Rio, who has very efficiently managed the discussion and deliberation. And in fact, he has generated the interest in all the participants. Because I see more than 400 participants are still there on this Zoom, and I'm sure on the YouTube also. So thank you, Dr. Rio, for taking out of time your busy schedule. We have been following your emails. So thank you so much. Then I would like to thank the speakers, particularly Dr. Hiroshi Izura, Jimmy Botella, for an excellent presentation, which is evidenced by the chat box that so much interest is generated in these two topics. And the eminent panelists who have been made it very lively discussion in the panel discussion, I would like to thank Dr. Jeba from Bangladesh, Dr. Tia Sharma from India, Dr. Kim from Korea, Dr. Hong from Taiwan, and Dr. Chan from Malaysia. And more than that, I would be very thankful and very grateful to all the participants who have turned up in a large number. As Dr. Vibha has mentioned, about 2,000 participants have registered. And I'm sure more than 60–70% have been attending this webinar. And this is not only the webinar end of here, as has been mentioned by Dr. Ravi in the morning and Dr. Vibha, that it is a series. So there will be 3 webinar series will be there. And before that, I make a request to you. I would like to thank profusely the steering committee, which have been talked about by Dr. Vibha, Dr. Ravi in the morning, who have very lucidly chalked out and guided us to develop these programs, to develop the resource document. And then the announcement, what I want to make, that we will be having the next webinar on advancing genome edited plants for Lab to Lent. And the date will be 4th August 2021. The registration link will be the same. And you will be finding very exciting speaker again, like you had today. There'll be Donald McKenzie from USA, Peter Thygesen from Australia, Florida Carino from Philippines, and the global panelists also from Philippines, Malaysia, and India. So stay tuned till 4th August. You enjoy your work and stay safe. Thank you so much, everyone, and bye-bye from my side. Bye.
Thank you.
Thank you.
Thank you, sir. Thank you
We have
so
a
much.
group photo, Rishi? No? Do we have a group photo?
I think, yes, Jack or Nui must be there. I think I would request them to click quickly and request to all of you to open your video so that we can capture your beautiful faces for our record. Just when you finish, please inform us.
Okay.
Okay.
Let's do the first one. 3, 2, 1. Okay.
Can you succeed?
Give me a second.
Okay. Okay.
Okay, let's do the second one quickly.
Okay.
Okay, 3, 2, 1. Smile.
Yeah.
Okay, thank you.
Thank you.
Thank you so much. See you on 4th of August to the very exciting webinar number 2, Advancing Genome Editing Plans from Lab to Land. Thank you so much.
Thank you.
Thank you.
Thank you, Richie. Thank you, Jimmy. Thank you, Heroes.
Thank you.
Thank you very much.
Thank you, Dr. Vibha, Dr. Rishi, for organizing.
Thank you. Thank you all.
Thank you.
Thank you, Rishi.
Thank you.
Yeah. Goodbye.
Bye.
You all.